In this tutorial, we will discuss about the gene expression, protein synthesis, transcription, translation, and central dogma. Yes, in our this single lecture, we'll tell you people about all these topics in a very easy way. The reason behind is these all are actually interrelated and interconnected. So let's start our very first topic, that is the gene expression. First of all, let's know what gene expression actually the term is. Gene, it is actually the fragment of DNA that has information. We have DNA and we have the fragment of DNA. So this DNA fragment, if it has information, then this is actually known as gene. Got what is gene? Simple. A fragment of DNA that has information. Now what is expression? Expression means to express. Now what is meant by expression here? Gene expression, simple. If this DNA has a fragment and that fragment has information for a specific function, here according to this lecture, our this fragment is going to do the synthesis of the protein. So like this information is expressed in the formation of what? Protein. So like this, if any information of the DNA is expressed in synthesis of something, that is actually called as expression. So here our gene expression is actually protein synthesis. Okay, protein synthesis. Now how the protein is synthesized, another topic. This protein is synthesized by means of transcription and translation. First of all, let's know what is transcription. Very simple and easy. Here we have the DNA. From this DNA, if this RNA is synthesized, which one? Messenger RNA, which has code for a specific protein, not code, codes, called as codon. So the synthesis of the messenger RNA from the DNA is actually called as transcription. Now, this is called transcription. Let's elaborate this a little bit more, that how actually this messenger RNA is synthesized. Very simple. We actually have 1, 2, 3 very important phases in the synthesis of this messenger RNA. The phase number 1 is now is initiation phase. In this phase, the RNA polymerase enzyme will find the promoter site on this DNA. Consider this is the promoter site on the DNA. As this polymerase, RNA polymerase, finds this promoter site, this will unzip this promoter site like this. And this will produce a bubble. This bubble is known as transcription bubble. And now these strains which are separated or unzipped by means of RNA polymerase, they are given the names antisense strained and sense strained. This antisense is going to be the template for the messenger RNA. So what we mean by the template? Simple. By means of this antisense strain, our RNA will be produced. How? Like means synthesized. So how now our RNA will be synthesized? Here we got the nitrogenous bases which are now free for the new available nitrogenous bases. So what will happen? Surrounding nitrogenous bases will come and will start binding to this free antisense template strained, antisense strained. Okay, so what will be now nitrogenous bases sequence? Simple. Adenine, cytosine, uracil, guanine, etc. So like this. So like this, these nitrogenous bases will start binding to the nitrogenous bases of the template strand, or you can say the antisense strand. Now this binding is actually known as elongation phase, when there is actually the elongation of the synthesis of the messenger RNA. Now this elongation is supposed to be terminated somewhere. Now what is the termination point in this elongation where the messenger RNA synthesis will get complete? That point observed is actually the guanine cytosine linkage. And this at this point there will be a kind of structure formed known as hairpin structure, GC hairpin, GC hairpin. Now this hairpin is actually the pin that is actually used by the females to hold their hair. Anyway, so this structure will be just like the hairpin. Somehow this, like this structure is observed, hairpin structure, where there is actually the GC observed, means guanine cytosine is actually paired at that point. So this is actually the point where the termination will happen of this RNA synthesis. After that, what will happen? The messenger RNA will be synthesized in the direction of 5 to 3. Now this synthesized RNA, it is not completely ready to go and come out to the cytoplasm to produce or to undergo other step. You can say to undergo the process of translation. What else is needed here is post-transcription modification. Now the transcription has already been completed. Now we need post transcription modification. Now this step is must for this newly synthesized messenger RNA. Why? Because as this messenger RNA is synthesized, as if this in the same style moves out from the nucleus into the cytoplasm, here we have phosphatase enzymes which are supposed to lyse our this messenger RNA. So in order to protect this messenger RNA, this newly synthesized messenger RNA from those phosphatases, nuclear phosphatases, it is must to be modified. So now how this messenger RNA will be modified? Very simple. At its fifth end, there will be actually the addition of 7-methyl guanosine triphosphate. This is actually a cape that is going to be placed on this fifth end of the messenger RNA, newly synthesized messenger RNA. And at the third end, there will be the addition of poly A tail. So poly A means several AAA. We have adenine, 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 1, 2, 3, like this. So there will be the addition of poly A tail. Poly A tail plus 7-methyl guanosine triphosphate, they will be added on this particular messenger RNA. Along with that, now we also need a proper code. So sometimes there is a kind of anti-code strand added in this messenger RNA. In order to remove that non-coding region from this messenger RNA, there is enzyme known as spliceosome that will then split those non-coding regions. Now we have actually one, two particular regions, segments, fragments, you can say segments in the particular RNA. Now this RNA has number of segments. Those segments are actually of two types. Segment number one is known as introns, number two is known as exon. Intron is actually, this segment is known as non-coding segment, whereas exon is actually the coding segment. So what spliceosome will do, they will split the introns because they are non-coding. So like this, the introns will be split, only the exons will remain, which are actually the coding segment, and we need those coding segments because this is the only segment that is responsible to produce the particular protein. So like this, now our messenger RNA is ready and having a proper code for the synthesis of proper protein. So now how the protein will be synthesized by this messenger RNA? First, the messenger RNA will move out. Now here, the messenger RNA is available in cytoplasm. So as it enters the cytoplasm, it will find the ribosomes. And you guys know that we have ribosomes, and we know about the ribosome that they are actually the protein synthesis factory. Means they're responsible to produce the protein. How? Like, before we move towards the synthesis, let's know the steps of this particular formation of the protein. The very first point you must remember is the translation. The formation of the protein from the messenger RNA is actually called as translation. First, coming to the point again, transcription is actually the formation of messenger RNA from the DNA, and translation is the formation of protein from the messenger RNA. When the messenger RNA is translated, then actually the protein is formed. So now how this translation is actually done? It is actually done by means of the initiation, elongation, and termination. Means in three phases the same translation is going to take place, just like the transcription. So now what is the initiation phase? First of all, messenger RNA as it enters the cytoplasm. Now according to messenger RNA, we got the concept earlier that it is synthesized in 5-3 direction. As it is reaching the ribosomes, its fifth direction, fifth end will bind to the small unit of the ribosome. After binding, what will happen? Then the large subunit will come. This is very important, simple point, that our ribosomes are actually composed of two subunits. One is small, another one is large. So small and large subunits will combine together. After that, they will accomplish, or they will do the synthesis of the particular protein. Now, what will happen is this messenger RNA binds by means of its fifth end to the small unit of the ribosome. In the meanwhile, the large unit will come and will bind to the small subunit of the particular ribosome. So now this here we got the ribosome, which has a messenger RNA in between. Messenger RNA has actually codon. Now, very important point. Codon will be of different types. So the one at the beginning will be known as start codon, and the one at the end will be known as stop codon. So here we have actually the start codon known as AUG, and the stop codon is UAA, UAG, UGA. Any one of these can be there. So these are actually the stop codons, and here we have the very first one, start codons, SRSP. Stop, SR stands for start codon. I just wrote it in short, don't be confused. So SR, start codon is AUG, and stop codon is actually UAA, UAG, UGA. Anyone can be here in this stop codon. Now this RNA is actually now available in between the small and large subunits of the particular ribosome. In the meanwhile, as they combine together, what will happen? Amino acids which are available in cytoplasm. Here we got the messenger RNA, they got the ribosome, so they are now attached with the ribosome. In the meanwhile, these amino acids, they will start activating. Their activation is actually done by enzymes. Amino acyl synthase plus ATP will be used. These will be activated, then they will bind to the transfer RNA. Come to the point. Amino acid, they will be activated by mean of amino acyl synthase enzyme, and this synthesis will use ATP also. So ATP plus amino acyl synthase, they will combine together, they will activate the amino acid. Now this activated amino acid is known as amino acyl amino acid. Okay, now this amino acyl will find the tRNA and they will bind with the tRNA. Now this tRNA which has got amino acid which is activated amino acid, this tRNA then it will be named as amino acyl tRNA. Now because of this amino acyl tRNA, here we got 1 2 3 regions in the large subunit of the protein. The very first region is now named as amino acyl region. Why? Because it is going to give the position or the space for the tRNA to come and to occupy that space. So now this tRNA is actually amino acyl tRNA. Because of this amino acyl tRNA, this first region will be named as acyl region. And now we shortly know this by A region. So now this A is actually from the name amino acyl region, which is actually for the amino acyl transfer RNA. I hope it's clear now. And the next point is the P region, where the peptide will be produced or synthesized. P stands for peptide. So here the peptide will be actually synthesized. Now the remaining point is the E region. This is actually the exit region from where the tRNA will be moving out. So tRNA will first will occupy the A site, where it will be having a specific amino acid. Which one? Amino acyl one, which is actually active one. Then it will come and it will be placed here in the P region. Here the peptide will synthesize. After that, this tRNA will move from the exit site. So like this, the tRNA will be moving through all these regions, and in the meanwhile, the peptide will be formed or will be synthesized. That's it, very simple. Now let me tell you people these all steps once again here in short. What will happen? We have the codon and anticodon on which? On the tRNA. Remember, tRNA is having the anticodon and this messenger RNA is having codon. These codon will be actually a kind read by the anticodon. So what will happen? They will read, and the meanwhile there will be the synthesis of the peptide. Now the very first amino acid which is going to be on the P region is actually the methionine. Methionine will bind to the P region. This is actually brought by mean of the tRNA. First of all, the tRNA will come at the A region, amino acyl region. Then from this, it will shift to the P region. In the P region, it will place the very first amino acid, that is the methionine. And then what will come? In the meanwhile, the next will be coming just after this particular, the very first amino acyl transfer RNA. Now the next amino acyl transfer RNA will be coming with the new and next amino acid. Like this, that RNA will enter the A, and then it will come to the P side. As that enters the P side, that tRNA will also place another amino acid here. So like this, the one that was on the P side RNA, that will be pushed by this previous tRNA to the E side. Now actually two regions are occupied, the one at the P and the one at the E side. Here we have tRNA, here we also have tRNA. Now the third tRNA, when it enters this A region, it will then push this one, this tRNA, then that tRNA will move to the E, and then the one that was at E region, that again will be pushed by the one which was in the P before. So like this, the new tRNA will be pushed out, means it will have an exit from the E site. So like this, the tRNA will be coming, and everyone will be placing a particular amino acid at the P region. So like this, the peptide will be formed, due to which this P region is named as P region because of the peptide synthesis. Now what will happen? A point will come when the stop codon will reach. Now as the stop codon reaches, then the synthesis will be stopped. So in the meanwhile, this produced polypeptide is free. This is now as a functional protein. So like this, the protein is synthesized. Now the remaining point is the central dogma. Now what is central dogma? Very simple. You got the concept from the DNA, the messenger RNA is synthesized. Then from this messenger RNA, if the protein gets synthesized, this is actually called as central dogma. Simple. The synthesis of the messenger RNA from the DNA and then the synthesis of the protein from this RNA is called as central dogma. Once again, DNA to RNA, RNA to protein is actually termed as central dogma. That's it from my side all. If still you have a question, drop that in the comment box, and thank you for watching.